Welcome back, class 9 to the online bio classes. We'll continue with the same chapter number 6, respiration in plants. We had already studied 2 experiments based on respiration. Today we are going to study the third experiment, that is to show that seeds take in oxygen during respiration. You know that oxygen is one of the important gas needed during respiration. And how can we prove it? With the help of an experiment, we need to take a conical flask. You know the conical flask? Okay. This conical flask contains a wet cotton on which some seeds are kept. The seeds are germinating seeds. They are germinating. Okay. And it has been stopped with the help of a cork. And then one bent tube, glass tube, has been inserted in it. And this bent glass tube is connected to a beaker, and that beaker contains water in it. Okay, that beaker is there. In that beaker, what is present? The water is there. Like suppose this much water level is present. Okay. This is setup number A, in which this is the germinating seeds. In your book already given, germinating seeds are there. And cork is there. Now you can find that one more is small, with the help of a thread, a small test tube is suspended. In that test tube, what is there? A chemical is there. The name of that chemical which is mentioned is soda lime, that is KOH, potassium hydroxide, or soda lime is there. Now one more experimental setup similar to this one has been set up here. But in this conical flask, instead of taking the germinating seeds on that wet cotton, we are taking the dry seeds, the seeds which are already dead, or the one which is not germinating. So dry seeds have been kept there, okay, as a control. Dry seeds are there, and then the similar experimental setup is there. So here cork will be there, and then this one is the bent glass tube which is connected to a beaker containing water is there. Alright, similar like that only. Previous one, water is also present here. Now what you will find, that after some time the level of water in this flask number A, in beaker number, this experimental setup number A, the water level will be drawn up. It will rise. Why it will rise, or why there will be increase in the level of water? It's because these germinating seeds, since they are germinating, so they need oxygen. So they will take oxygen from the surrounding, that is from the flask. As a result, now what is the function of that KOH which is kept there? Water rises in the bent tube. In this tube, the water will rise up. Why? Why the water is rising up? It's because the oxygen gas which is being used up during respiration, it creates a vacuum or empty space, and it draws the water molecules upward into this bent glass tube. Now what is the function of that KOH which is there in a small test tube? KOH solution is there in order to remove or to absorb the carbon dioxide gas which is being produced by the process of, like during the process of respiration, one gas called carbon dioxide gas is also produced. So if you take a small test tube containing soda lime or KOH solution in it. Now what is the function of that one? Carbon dioxide produced was absorbed by the soda lime. Its function is to absorb, already I said, to absorb CO2. Now from where does this CO2 comes? It comes from the germinating seeds. So you will find that the level of water rises in the bent tube in experimental setup number A, but in setup B there is no changes in the water level. It remains same only up to where it was present in the beaker. So from this experimental setup we conclude that oxygen is the one which is produced during germination of seeds. Okay. Now next we have to study is types of respiration. Depending on the presence or absence of oxygen gas, there are 2 types of respiration. If respiration occurs in the presence of oxygen, it is called aerobic, or the word which is used in a book is oxybiotic respiration. Whereas that type of respiration which occurs in the absence of oxygen, it is called anaerobic respiration, or the word which is used is anoxybiotic. So oxybiotic is the other name of aerobic respiration, same like aerobic respiration. And the other word which is used, that is anaerobic respiration, anoxybiotic. So both of them are same. Anoxybiotic is also known as anaerobic respiration. That is, the respiration which takes place in the absence of oxygen is called anaerobic respiration. Now you know that during aerobic respiration, due to the presence of oxygen, there is complete oxidation or breakdown of glucose molecule, and it will produce large amount of energy, that is 38 molecules of ATP is also produced. But during anaerobic or anoxybiotic respiration, there is incomplete burning or oxidation of glucose molecules, and it will lead to very less amount of production of energy, that is only 2 ATP molecules are produced during anaerobic respiration. Okay, so what is the equation of aerobic respiration? If we need to write the respiration, anaerobic respiration and aerobic respiration, so first I will write the equation, chemical equation, which is mentioned in your book also. The chemical equation is C6H12O6, that is glucose molecules, when it combines with 6 molecules of oxygen in the presence of enzymes. Respiration occurs in the presence of enzymes. It is a stepwise process and a slow process. It results in the formation of 6 molecules of carbon dioxide plus 6 molecules of water, plus that energy which is produced is 38 molecules of ATP is produced. So energy comes in 2 forms. What are they? This is the ATP, that is the energy of the cellular respiration, and the second energy is heat energy, which is produced in the animals particularly. So this much amount of energy is produced: 6 molecules of carbon dioxide, 6 molecules of water, and 38 molecules of ATP generated during aerobic respiration. So this is the chemical equation for aerobic respiration. Now anaerobic respiration, also called anoxybiotic respiration, it occurs in the absence of oxygen, and generally it is found in our skeletal muscle cells. In plants also they respire anaerobically, and the equation which is present for anaerobic respiration is C6. The substrate is same. What is the meaning of substrate? The substance on which respiration occurs. So substrate is same, that is glucose, C6H12O6, that is glucose. Now we no need to write here 6O2 in case of anaerobic respiration. Directly in the presence of an enzyme, the name of the enzyme is mentioned in your book, it is gymase enzyme, Z-Y-M-A-S-E. Gymase enzyme is required for anaerobic respiration. It produces 2 different products. Now in your book what is mentioned here is ethyl alcohol, 2 molecules of ethyl alcohol. So the formula of ethyl alcohol is C2H5OH. OH is the hydroxyl group which is found in alcohol, so it is called ethyl alcohol. Why? Because C2 is there, ethene, from ethene it is formed, so it is called ethyl alcohol. So it happens that this ethyl alcohol is the product of anaerobic respiration in plants only. In case of animals it is different, it is called lactic acid. Apart from that, 2 molecules of carbon dioxide is also produced, and how many molecules of ATP is formed? Only 2 molecules of ATP is produced during anaerobic respiration in plants. Now next one is, what is respiratory quotient, RQ? Respiratory quotient is the ratio of the amount of carbon dioxide produced to the amount of oxygen which is used during the process of respiration. In your book the definition is also given there. Relation between the volume of carbon dioxide. So if we have to write the formula RQ, RQ stands for respiratory quotient. It is equal to volume, I am not writing the full word, volume of CO2 produced during respiration by volume of oxygen used during respiration. So when you get a ratio, that ratio is called respiratory quotient. Got it? What is the meaning of respiratory quotient? It is the ratio of volume of CO2 produced by the volume of oxygen which is used during the process of respiration. Now next, germinating seeds. During the early stages of their germination, they respire anaerobically. Why? Because their seed coat is very hard and they will not allow the diffusion of oxygen gas which is needed for the process of respiration. Now next we have fermentation. Certain microorganisms means those living. Okay, now next one is, what is this fermentation I was saying? Yeah, fermentation is a type of anaerobic respiration which is found in microorganisms like bacteria and fungi. They respire anaerobically, and that process of anaerobic respiration in microorganisms is called fermentation, and this process is utilized in the manufacture of bread, cakes and breweries also, where the alcoholic products are made. Now there is the fourth experiment, that is to show anaerobic respiration. So I will just show you with the help of a diagram. Diagram is already there in the book also. It's very simple experiment. What we have to do is, we have to take a stand, and then with the help of a clamp, this is called a clamp, we have to take an inverted funnel or a cylinder which is filled with mercury. What is filled inside? Mercury is filled. And here some seeds are kept, germinating seeds are there. Okay. Now a similar experimental setup will be taken. This is experimental setup number A. The second one is number B. Germinating seeds without seed coat. This one is seeds without seed coats. Seeds without seed coat. There is no seed coat in it. Covering is not there. Okay. The one which is germinating. Now in experimental setup number B, we take another experimental setup. Similar apparatus will be used, and what we will find that here also mercury is filled, but in this case the seeds which are present, they are without seed coat. Here also it is present. Yeah, germinating seeds without seed coat is present. Now you will find that a lot of gap or space is produced here. Here in this case there is no gap. The mercury is completely filled and the seeds are kept in it. Okay. Now what does this experiment show? From this experiment, you have kept some germinating seeds without seed coat in both the tubes, test tube A and B, inverted it and sealed this one and filled mercury inside it. You will find that after some time a vacuum is created here in the upper part of the test tube. That vacuum contains a gas which is CO2, carbon dioxide gas. Now from where is that carbon dioxide gas produced? It is produced by the germinating seeds due to anaerobic respiration, means they are respiring in the absence of oxygen. So what is the conclusion? Carbon dioxide which is given by the seeds during anaerobic respiration has lowered the mercury. Why it has pushed the mercury down here? Because CO2 gas is filled inside it. From where does it came? It came from respiration, anaerobic respiration of the germinating seeds. Okay, it has lowered the mercury level, and with the addition of KOH, if you add potassium hydroxide here, KOH, once again the same condition will be there, like the level of mercury will rise up. Why? Because the function of KOH, when it is added, soda lime is added, it will absorb that carbon dioxide gas. Means now the vacuum which is created will be filled by the rising level of mercury. Got it now? Now next is differences between anaerobic respiration and fermentation. Tabular differences is given. I will just say the differences. First one is anaerobic respiration. The substrate glucose is respiring in anaerobic respiration. In fermentation, the substance which undergo respiration is extracellular medium, not the one that is glucose which is present inside the cells. Number 2, anaerobic respiration occurs in the living things inside their cell. It is called intracellular respiration or intracellular process, whereas fermentation occurs outside the cell. It is called extracellular process. The third one is end products of anaerobic respiration occurs inside the living cells, whereas in fermentation the end products are found in the outside medium, not inside the cell. And the last one, number 4, anaerobic respiration causes foaming, that is the froth is formed, whereas fermentation will not produce any type of foaming. Foaming will take place in fermentation, it will not take place in case of anaerobic respiration. So write these tabular differences in your copy and learn it. Then next we have differences between aerobic and anaerobic respiration. First difference you can say, aerobic respiration occurs in the presence of oxygen, anaerobic respiration in the absence of oxygen. Second difference, aerobic respiration produces large amount of energy, that is 38 ATP. Anaerobic produces less amount of energy, that is 2 ATP. Third difference you can say, aerobic respiration there is complete oxidation of glucose molecules. Anaerobic there is incomplete oxidation of glucose molecules. Fourth point you can say, aerobic respiration there is no formation of ethyl alcohol or lactic acid, but during anaerobic respiration ethyl alcohol is produced. And fifth difference you can say, aerobic respiration occurs throughout life, means till a person or a living thing is alive, till that time aerobic respiration will continue, but anaerobic respiration occurs only for a short time, for a temporary period of time. Okay, and next we have the section exercises. You can try and complete those section exercises as homework. Next we have to continue is the next topic, factors affecting respiration. In the next class we can continue with that, factors affecting respiration. Meanwhile the homework will be to write both the tabular differences in your copy and learn it, and all the different types of experiments which are there. Just go through the experiments, read it, try to understand. If any doubt you can ask me when we will conduct the next interactive classes. So in the next class we'll do the interactive class. Be ready for that. Okay, thank you.